Hello, this is Susan Knowles from Cochise College, and welcome to this podcast, Introduction to the Cardiovascular System Conduction. During this podcast, I will discuss the conduction pathway, the normal conduction pathway for the heart, as well as both the normal sinus rhythm waveform and two life-threatening ECG waveforms. The normal pathway for the conduction of the impulse through the heart is to begin at the sinoatrial node, which is the SA node. The SA node is located at the upper part of the right atrium, sort of near the location where both the inferior and the superior vena cava dump into the right atrium. The impulse begins there. It travels down to the AV node, or the atrioventricular node. There it slows down for just a moment to allow the atria to completely fill with blood, and we'll talk about blood flow in another podcast. And then the impulse will continue down through the bundle of His. It splits into the right and the left bundle branches, going down into the ventricles and finally down to the Purkinje fibers. It's a combination of the impulse as it passes through the bundle of His, the right and left bundle branches, and the Purkinje fibers. Together, their impulse is what actually causes the ventricular contraction. The heart can contract by itself, independent of any signals or stimulation from the body. The heart contracts in response to an electrical current conveyed by the conduction system. Just to reiterate, specialized cells in the SA node near the superior vena cava initiate an electrical impulse. Because the SA node has an intrinsic rhythm, it is the pacemaker. The current flows in an orderly sequence, as I said, first across the atria to the AV node low in the atrial septum. And there it is delayed slightly so that the atria have time to contract before the ventricles are stimulated. Then the impulse travels to the bundle of His, the right and left bundle branches, and then through the ventricles. The electrical impulse stimulates the heart to do its work, contract. A small amount of the electricity actually spreads to our body surface where it can be measured and recorded, and this is what is recorded on the ECG or the EKG, whichever way you want to word it. And the ECG waves are arbitrarily labeled P, Q, R, S, and T, which we'll talk about in a few minutes. Conduction pathways are the expected paths which electrical activity should take as it travels through our heart. Unlike other cells in the body, myocardial cells can create and conduct electrical impulses, and this is called automaticity. As we expect that the electrical impulses will be created by the primary pacemaker sites, not every other myocardial cell, and that the electrical current should take the expected usual pathways. The primary pacemaker sites are our SA node, and it is responsible for the atrial contraction. And it has an intrinsic rate or a preset firing rate of 60 to 100 beats per minute. And as long as the SA node is firing correctly, all the other sites will not. It's very important. Other nodes of the heart monitor the heartbeat, and if the heartbeat drops below 50 beats per minute, the AV node will take over pacing the heart. And the AV node is called the gatekeeper because it does watch the rate of the heart as it comes down from the SA node. The AV node's intrinsic firing rate is between 40 and 60, and its main function is to regulate the impulse from the SA node. The AV node under normal conditions will slow the heart down, or the current, before the signal passes through to the ventricles. The delay ensures that the atria have a chance to fully contract before the ventricles are stimulated. If the impulses are coming too fast, then the AV node selectively allows only some of those impulses to be conducted, and this is how it gets the name as the gatekeeper. If the impulses are too slow, then the AV node can generate the impulses at its own intrinsic rate, as I mentioned, somewhere between 40 and 60 beats per minute. So if we take a patient's pulse and it's 46 to 48, then perhaps the AV node has become the pacemaker. The AV node then conducts impulses to both the right and left, first through the bundle of His, and then the bundle of His carries the impulse from the AV node to the right and left bundle branches and eventually to the Purkinje fibers. And as I've said before, the bundle of His is actually named after the person who discovered it. The right and left bundle branches and the Purkinje fibers all work together and are responsible for contracting the ventricles. The passage of the electrical impulse along these three pathways causes the ventricles to fully contract. The cells of the ventricle have their own intrinsic rate, and it's between 20 and 40 beats per minute. The ECG should have a smooth baseline and distinct complexes. Atrial and ventricular depolarization and repolarization are electrical events that are seen on the ECG. And remember that depolarization is actually contraction or systole, and repolarization is relaxation or diastole. The P wave represents the depolarization or contraction of the atria. A normal PR interval is a normal impulse conducted from the SA node in the atria through the AV node. It means that it made contact with the atria on the way down, and it followed a regular, unobstructed course. On the EKG, it is the time it takes for the electrical impulse to start at the SA node, be conducted through the atria and the AV node. It is measured from the beginning of the P wave to the beginning of the QRS complex, and it represents depolarization of the SA node and the atria down to the AV node. The QRS complex represents the depolarization or contraction of the ventricles. The ST segment is the area between the end of the S wave and the beginning of the T wave, and it should rest on the line. The T wave represents the repolarization or relaxing of the heart, and it is more rounded and larger than the P wave. So what is the definition of normal sinus rhythm? Normal sinus rhythm means that the impulse followed a normal path of conduction from the SA node to the AV node to the bundle of His to the Purkinje fibers. The tracking is of a normal rate between 60 and 100 beats per minute, and the rhythm is regular. Should you see a prolonged PR interval, that would mean that there was some delay in the conduction of the AD through the AV node. And on the next slide, or one after that, the next slide, I think that one we will be discussing the EKG paper so that you will be able to see what the expected or reference range, if you want to put it that way, of each of the ECG waves forms of the complete complex. This is the way we'll be able to figure out whether or not the conduction of the impulse was within the normal limits. So the ECG complexes measure the electrical events, whereas the paper measures the time. The ECG paper rolls out of the machine at a preset speed of about 25 millimeters or 1 inch per second. The paper is marked off in a grid with each line 1 millimeter apart, so every fifth line is darker and thicker so that the very small boxes can be counted accurately. Each small box represents 0.04 second. Each large box is made up of 5 small boxes and represents 0.20 second. So you can see that the EKG paper represents a very short period of time. The electrical event that takes 0.08 of a second would indicate, or create a line between 2 small boxes long. So let's talk about what's normal. A P wave is 3 boxes, or 0.12 second wide. A PR interval would include the P wave to the beginning of the R wave. But it actually looks like it kind of only goes to the Q, so don't get confused about that, but that's what's considered the PR interval. And that represents about 3 to 5 small boxes, or 0.12 to 0.20 second wide. The QR complex, the normal time would be 0.04 to 0.10 second, or 1 to 2.5 boxes wide. The height of the QRS is adjustable on each machine, so that's not necessarily something that you're going to be concerned about when you're evaluating an ECG strip. The T wave is upright, it's well rounded, and it's less than half the height of the QRS complex. The ECG is a graphic representation of the electrical activity in the heart. So by counting the boxes, we can establish the time the complex takes to travel. By counting the number in 1 minute, we then can establish the rate. If the ECG complex is wider than normal, it means that the electrical impulse was slowed somewhere in the heart. And if it's more narrow, then it means it is traveling faster. If it has a strange shape, then the impulse was rerouted somewhere through the heart. So that's why I've been emphasizing normal conduction pathway, so that when you look at an ECG pattern or a waveform, and you see if it looks like what we consider to be normal sinus rhythm, then you know the heart has taken this normal pathway. However, if it looks bizarre, then we know that the impulse was rerouted somewhere. So we're going to go ahead and talk about ECG rhythms that originate in the SA node. So what do I mean by that? I mean a rhythm that actually takes the normal conduction pathway because it begins from the SA node. Any rhythm that begins in the SA node will have a P wave because the P wave represents the conduction from the SA node down to the AV node. So when you start to look at ECG monitoring or waveforms, you look to see if it has a P wave. If it has a P wave, you know that the conduction began in the SA node. So we're going to be looking at normal sinus rhythm, sinus bradycardia, and sinus tachycardia. This is an example of EKG paper. It's enlarged so that you can see where the small boxes are, what would be considered a large box. You see the ECG waveform, the P, the PR interval, the QRS complex, the ST segment, as well as the T wave. You can ignore the U wave at this particular time. So as you go farther in your education for the cardiovascular system, when we hear about ST segment elevations or depressions, or when you talk about different aspects of the ECG, these different divisions, if you will, of the ECG will make more sense, but we're not going to be too worried about all of that at this point. We're just looking to compare what normal sinus rhythm looks like compared to life-threatening rhythms, and whether or not it's an atrial rhythm or it's a ventricular rhythm. An atrial rhythm will have a P wave. A ventricular rhythm will not have a P wave. A ventricular rhythm is not going to be a normal ECG, because I've just explained to you that we want the impulse to begin at the SA node. So we need to have a P wave. And sometimes some patterns don't. So the easiest, most accurate way to calculate a strip is by rounding the number of complexes in a 6-second strip rather than running a full 60-second strip. So you run a 6-second strip, and you can tell that by the number of large boxes on the EKG paper, and then you would multiply it by 10, and that's what you would have it rounded in 1 minute. So if you looked at this particular example on the screen and you counted the number of QRS complexes, you would see that the rate is 70. It is between 60 and 100, so you know that's the appropriate rate for the SA node. If you look at it, you will notice that there is a P wave. So that means that it is a sinus rhythm. When you look at it, you'll notice that the rhythm is regular. You'll notice that each QRS complex is identical. The PR interval falls within the normal range, as well as the QRS. So we now know that this is normal sinus rhythm. Here's yet another example of normal sinus rhythm. So if you look at this EKG paper, you'll notice that it has some spikes that run off of the paper. So what you want to do is you want to make sure that you are actually looking at 6 seconds of the paper. So in between, so at the beginning of it to the far left, as you're looking at the screen, you can see that there is a complex there, but you wouldn't count that complex within your 6 seconds. You'll also notice that at the far other end of the strip, that is not part of within the 6 seconds. So make sure you're only counting the number of complexes within 6 seconds. So if you look at this strip, you will be able to count the number of complexes. So you go ahead and do that for yourself. And when you do that, you'll come up with a number. You'll find that the rate does fall within the 60 to 100 beats per minute. So that is within normal sinus rhythm. You'll look at it and you'll notice that each complex is complete. It does include a P wave, which makes it sinus. It does have a QRS complex as well as a T wave. There's no wild or bizarre looking complexes. All the intervals are regular and they appear to be normal in appearance, so this is considered normal sinus rhythm with a rate of 100 beats per minute. Our next example is going to come from either being tachycardia or bradycardia. Sinus bradycardia. Well, if we once again use the same method to evaluate the strip that you see on the screen, you want to make sure that you're only counting the number of complexes that fall within a 6-second period of time. Once you look at that, you want to evaluate it systematically. Is there a P wave? Is there a QRS complex? And is there a T? If so, you know you have complete complexes. Because there is a P wave, you know you have sinus rhythm. Because you have a QRS complex, you know you have a ventricular contraction. There are no weird or bizarre looking complexes. And all the intervals, except the rate, are within normal limits. So when you count the number of complexes here, you'll note that there's 1, 2, 3, 4 complexes. So it looks like the rate would be 40. It makes it in the bradycardia range because it's less than 60 to 100. But we do know it's still a sinus rhythm because we have a P wave. So this patient probably is pacing from the AV node, but it's still within the atria. So this is considered sinus bradycardia. So to contrast our previous strip with this strip, you'll note that once again, go through the same process. Make sure you're only counting the number of complexes within a 6-second period of time. We note that the rate is much faster than it was on the previous example. We will count the number of QRS complexes. We'll make sure that they seem to be regular in appearance, that there's a P wave, that there's a QRS complex, that there's a T wave. There's nothing weird or bizarre about it, except that it is faster. And the only thing that falls outside of within normal limits for normal sinus rhythm is the rate. So that we know that the rate on this one is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Looks like it's 140 beats per minute, which makes it tachycardia. And it does have a P wave, so it initiates in the atria. So this is sinus tachycardia. So next we'll talk about a couple of life-threatening rhythms, VFib or ventricular fibrillation, and asystole. Ventricular fibrillation is the beating of many weak ectopic foci in the ventricle. So what does that mean? It means that ectopic means that it's outside of the normal conduction pathway, that the cells are just quivering. And because of that, it's an uncoordinated, undulating, instead of a coordinated contraction. It's a rhythm that cannot circulate blood, and it's not compatible with life. So if you were trying to count the number of beats per minute per se, it's uncoordinated. It's not firing properly, and you can't really count the rhythm. It's a deadly rhythm. If help isn't gotten right away, the patient will die, and it's quite possible the patient will die anyway. Asystole. This rhythm is associated with death. It really isn't a rhythm. Asystole means without ventricular contraction, because you now know what systole actually means. So asystole is a flat line. It's not a rhythm, but the absence of all electrical activity. And unless swift or sure interventions take place, this will lead to death. And as I said in the previous rhythm as well, if prompt medical attention is not delivered, the patient will die anyway. So I want to thank you for joining me on this podcast. In class, we went about trying to evaluate some of the same patterns we've talked about in this podcast. But remember to use these guidelines. What is the rate? Is each complex complete? Is the R to R interval constant? Does any complex look wildly abnormal? And once you've answered those questions, you'll be able to go about naming the rhythm. Thank you. This is Susan Knowles from Cochise College.